welcome to Medical Dialogues. I'm Mr. Zaman and here's what I bring to you all from the world of medicine.
Alzheimer's Diagnosis New Guidelines. Recently, at the International Alzheimer's Congress in Amsterdam, new guidelines for diagnosing Alzheimer's disease developed by clinicians and researchers from around the world were presented. In these criteria, the disease is diagnosed in the clinic through the use of blood biomarkers, just as with other major diseases such as diabetes and cardiovascular disease. New research shows that many people with cognitive symptoms want to know whether they are in the early stage of Alzheimer's. A diagnosis enables patients to take more control over the next phase of their lives. The desire to know whether or not you have Alzheimer's disease makes the use of blood biomarkers so relevant. It is also the gateway to treatment. The Swedish research presented at the ADPD conference in March has shown that biomarkers can be more reliable than the analysis of a primary care physician. Bacterial protein in urogenital tract linked to fertility issues and birth defects. A team of researchers from the University of Maryland School of Maryland's Institute of Human Virology published new findings that emphasize the crucial role of the urinary and genital tract microbiota in adverse pregnancy outcomes and genomic instability that originate in the womb during fetal development. The study established a new link between genomic instability and a protein from Mycoplasma fermentans, a kind of bacterium that commonly colonizes the urogenital tract. The bacterial protein also reduced fertility in mother mice and resulted in more birth defects in the newborn pups. For this latest study, researchers created mice that make the DNA K protein normally produced by the bacterium Mycoplasma fermentans. Understanding how the skin cancer virus outcompetes host cell replication. University of Pittsburgh researchers have shown for the first time how Merkel cell polyoma virus, MCV, which causes an aggressive skin cancer called Merkel cell carcinoma, initiates DNA replication in host cells. In a new study, the researchers studied MCV replication in never before seen detail using an instrument called a C-trap and a technique called SMADNE. During normal cell division, the first step of DNA replication involves proteins called helicases that form two sleeves around the DNA double helix. These sleeves push together to unzip the double-stranded DNA into single strands so that other proteins can bind and perform the next steps. This unzipping process requires cellular energy in the form of molecular ATP. Known as licensed DNA replication, this controlled process occurs only once each time a cell divides.
That's all for today. Stay tuned to Medical Dialogues for latest updates.
Never miss a medical update from Medical Dialogues. Like, subscribe, and press the bell icon.